Put your fingers to your neck or your wrist. Can you feel that subtle pulse? That pulse is your heart working. The heart is one of the most vital organs in the human body. The heart works to deliver oxygen and nutrients to every cell in your body by pumping blood. Cells need that oxygen and those nutrients to thrive. Cardiovascular disease is the #1 cause of death worldwide. One such deadly disease is known as coronary heart disease, and it's caused by excess cholesterol and the buildup of excess cholesterol in the arteries and veins of the human body. Cholesterol is a wax-like fatty substance that is produced by the liver. The body uses cholesterol in order to produce vitamin D and to help build cell membranes. Cholesterol is attached to the proteins on blood cells, which is how it gets transported through the blood. Excess cholesterol won't attach to these proteins and is left on the arteries, the inner walls of the arteries. This leads to the hardening or narrowing of arteries, which eventually leads to the complete stop of blood flow in the heart. This then leads to chest pain known as angina, which has been linked to causing heart disease. So what has already been done to try and treat coronary heart disease? A class of drug known as statins directly interferes with the production of cholesterol at the liver. Statins are an effective form of treating coronary heart disease, but they are not the solution. That is where gene editing comes in. Gene editing provides a relatively long-term solution. You wouldn't need to take a drug a day. The technology involved in creating this gene edit is called CRISPR-Cas9. CRISPR stands for clusters of regularly interspaced short palindromic repeats. And Cas stands for CRISPR-associated protein. The 9 is the number of the protein. The CRISPR-Cas9 system can be thought of as a pair of molecular scissors. Let's say you have a DNA sequence you would like to alter. The CRISPR-Cas9 has an RNA sequence that is programmed to be complementary to this DNA sequence. The RNA sequence acts as a guide for the Cas9 protein. Once the Cas9 protein is brought by the RNA to the site at which you want to have the DNA cut, it will cut the DNA at that site. CRISPR-Cas9 was specifically used to target the PCSK9 gene. Produced and expressed by the liver, the PCSK9 gene has been linked to an increased risk of coronary heart disease in some individuals. Specifically, it interferes with the receptors of cells for cholesterol, which thus inhibits the way they are able to take in cholesterol and use it. Scientists used CRISPR-Cas9 in order to target exons 1 and 2 of the PCSK9 gene in the blood cells of mice. And it worked! The rate at which this mutation was incorporated into cells was over 60%. And after 2 weeks, the cholesterol level in the blood cells of these mice went down almost completely. This has huge implications for the humans who have the PCSK9 gene. These people, thanks to clinical trials which are already being done in order to develop the drug that is based off of this CRISPR-Cas9 system, can look forward to a healthier heart. I plan to simulate this experiment by using the genome compiler software. This software will let me directly mutate the given exons in the PCSK9 gene and simulate assays, just like a researcher would in a real lab. Stay tuned for my report on this simulation.